morning. I believe that now we're ready to start our today's lecture. I hope that you're all having a good day, and I hope that you've managed to revise most of your lectures so far. So I will be waiting maybe over the weekend or over these days. If you have any questions, to email me so that we are able to solve any problems that you may be facing. I would like to thank you for all your emails and your constructive feedback on the lecture slides. There were some mistakes that I have not noticed, such as, for example, during the luteal phase, we said that inhibin is being released, although that's not true because we only have estrogen and progesterone being secreted during the luteal phase. I hope that all of you have seen the video that I've uploaded over the National Day, and that now you should not have any issues in regards to the hormones and the feedback cycles between the ovaries and the anterior pituitary. So today we will be talking about the influence of the ovarian hormones on the uterus and the endometrium. We will be speaking about the formation of the trophoblast, as well as the placenta and the hormones that are being exchanged between the fetus and the mother. We will be talking about parturition, and finally, we will be speaking a little bit about lactation. So overall, we will be covering approximately 60 slides over the next 2 hours. So please, I would like to have your attention because we'll be mentioning about some complicating issues. So, and hopefully during the end, we will have some time for questions and answers. So, throughout the ovulatory cycle, we've seen that there are two main hormones that play a significant role. We have estrogen and also progesterone. So these two hormones, they have a specific impact on the endometrial wall. And the reason is because that during this phase, the female reproductive system is preparing for potential fertilization and implantation of the zygote. So just to get familiar with the terms that we will be using, myometrium is the outer smooth muscle, which you should know by now through your anatomy classes. And the endometrium is the inner line that contains the numerous blood vessels, glands, as well as glucose that is responsible for the implantation. The ovarian cycle, as well as the uterine cycle, consists of 28 days, and it is separated in 3 different phases. We have the menses, the proliferative phase, as well as the secretory phase. So throughout this cycle, the endometrium is being prepared for implantation. Under the influence of estrogen, we have the growth of the myometrium as well as endometrium. And during this phase, estrogen is priming the endometrial wall for the influences of progesterone. We have, during this phase, we have the progesterone receptors that are being built up, whereas progesterone comes and tops up this process through in which we have the secretion of glycogen by the endometrial wall. We have the growth of the endometrial vessels, and also we have the accumulation of electrolytes and waters that is gonna facilitate the implantation. During the phase under the progesterone influence, the endometrium is particularly malleable and facilitating an easy and smooth passage for the newly formed zygote. In addition to this, progesterone is a muscle suppressant, thus it provides also a quiet environment for the newly formed life. So the first phase of the menstrual cycle is the menstrual phase. It is characterized by the end of the luteal phase and the beginning of the follicular phase. It takes approximately 7 days, during which, under the influence of, under the withdrawal and influence of specific hormones and the release of prostaglandins, we have the shedding of the endometrial wall. So as the corpus luteum degenerates, also progesterone and estrogen levels starts declining. During this phase, and since the hormonal environment is being withdrawn, in addition to the death of the corpus luteum, we have the release of the uterine prostaglandins. What happens during this phase, we have the suffocation of the blood vessels that have been developing through the estrogen and progesterone cycle, and eventually, under the reduction of oxygen, we have the death of the endometrial wall, as well as the vessels that are surrounding the endometrium. During this phase, and as we know, under the influence of prostaglandins, which are myocontractants, we have the myometrial shedding and the mild rhythmic contractions that would lead to the shedding of the endometrial wall that comes out in the form of menstrual flow. In the occasion that certain females have excessive levels of prostaglandins, then they may experience a form of pain, which in its clinical terms is called dysmenorrhea. During this shedding, still we have a thin layer of epithelial cells and glands that are remaining in the endometrium, which are gonna start proliferating in the next ovulatory phase. Menstrual flow never clots due to the fact that an enzyme is present called fibrinolysin, and the fibrinolysin enables the blood never to clot. In addition to this, we have the presence of leukocytes that inhibit any bacteria or viruses in causing any form of infection, due to the fact that at this phase, the endometrial wall is particularly vulnerable to bacterial infections. Now, moving from the menses to the proliferative phase, during this proliferative phase, our endometrium is under the influence of the estrogen levels. So estrogen stimulates the proliferation, as we've said, of epithelial cells, glands, and blood vessels, as well as, and most importantly, it induces the synthesis of progesterone receptors in the endometrium. During this phase, the proliferative phase coincides with the LH surge. So estrogen levels are high and LH levels, and under the influence of high LH levels, we're gonna be going into the mid-cycle phase where we have the release of our secondary oocyte. Now, following the proliferative phase, we have the secretory or progestational phase. Why secretory? Because under the influence of progesterone, we have the elevated release of glycogens in the uterine lumen, through which the morula, prior of implanting, is gonna feed for a period of 7 days. In addition to this, it is also called progestational phase because the endometrium at this phase, it's highly vascularized, it's filled with glycogen and minerals, which is gonna provide a fertile ground for the newly formed trophoblast. Some other changes that are happening under the influence of estrogen and progesterone is the cervical mucus. Now, the cervical mucus, as we said in our previous lecture, and under the influence of estrogen, it's very thin and it's also abundant and clear. That is, it enables the sperm to have a clear passage to the female oviduct for fertilization. In addition to this, under the influence of estrogen, we have what we call the ferning effect. Now, ferning, why ferning? Because it looks like a fern, the plant fern. I don't know if most of you must be familiar with Jurassic Park, that you see all these ferns. So the ferning effect that you see over here, it basically creates a smooth passage for the sperm to start crawling its way up to the newly released oocyte. Now, after ovulation, we have the change of the cervical mucus in becoming very thick and sticky. It more or less looks like during this phase, it looks like a candle. So it's very thick. Under this influence of progesterone, the sperm has much greater difficulty in passing through the cervical canal. And the reason is because during this phase, and because of the fact that the endometrium is rich in water and glycogens, it does not only provide the ground for the newly formed zygote for implantation, but it also provides a fertile ground for other bacteria that could threaten the valuable female reproductive system. Okay. Now, go away. Okay. Now, there are occasions that the female is deprived from estrogen and progesterone, and this happens usually during menopause at the age of 45 to 55 years old, where the female depletes its follicular deposits. Now, as soon as these ovarian deposits are depleted, then we have a climacteric period, or the perimenopausal period, in which it kind of informs the female that is going to go through this phase of estrogen or progesterone depletion. During this phase, the female may experience a variety of physiological changes, such as hot flashes, rapid elevation in temperature, due to the fact that rapid estrogen influx is also a very potent vasodilator. That's why usually perimenopausal or menopausal females, they experience these hot flashes, particularly in perimenopausal females. Now, as we said, estrogen is also responsible for the closure of the epiphyseal plates. In addition to this, and under the low estrogenic environment of the females, we do not have anymore the beneficial osteoblasts that are happening for the deposition of calcium and minerals in the female bones. However, under the lack of estrogen, we have what we call osteoclasts. So we have the phenomenon of osteoporosis. It is beneficial in females under menopause to go into hormone replacement therapies that eventually it's gonna diminish the adverse effects that are being experienced by the osteoporosis, as well as the psychological and physiological impact of the lack of estrogen. Now, moving from menopause to menstrual irregularities in female athletes, it has been recommended by a scientist the hypothesis of the minimum fat deposit or the minimum caloric deposit. So if athletes that are low on fat or low on calories, they will experience a form of menstrual irregularities that could come in the form of hormonal imbalance, or even at the worst case scenario, complete cessation of ovulation or amenorrhea. Now, because ovulation and let's say fertilization of a newly released oocyte is a costly process, the organism goes into a process of oocytic arrest. Thus, during this phase, we have a diminishment of estrogen and progesterone that eventually is going to lead not only to the endometrial buildup, but it's also going to suppress the release of newly formed follicles. In this way, the female not only protects this important genetic material, but it also does not undergo into this, let's say, costly reproductive process. Causes for this could, for the menstrual irregularities, could include rapid weight loss, decreased percentages of body fat, dietary insufficiencies, and also prior menstrual dysfunctions. Stress is also another potent psychological factor for these menstrual irregularities. So females could go into menses much earlier under periods of stress. So as we said, we have depressed FSH levels and LH levels, and in addition to these menstrual cycle irregularities, we could have other forms of physiological problems, such as potential fractures, due to the fact that the hormonal environment, particularly estrogen, is being disrupted in the female athletes. Then the mineral density, then osteoporosis could occur. The good news, however, with female athletes is because of the fact that estrogen levels are low, and since most of the cancers are responsive to estrogen, we have a 50% decrease in cancers in the reproductive system of the females, if that female particularly exercises. Now, exercise is particularly important for females. However, it should be performed under a specific diet and should also be performed in moderation. So anything excessive that happens in physiologically in our life is definitely going to affect us in an adverse way. So this brings us to the end of our first lecture. I don't know if you have any specific questions before I move on. Okay, so we have R&D new studies suggesting similar andropause, like menopause, but in later years. So yes, we do have andropause in later years. However, this is mainly caused by bad lifestyle habits, and this happens mainly because of vascular changes that are happening in the testes. It could be either because of excess fat deposit, it could be because of cholesterol, stenosis of arteries, atherosclerosis, and so on. So a lot of non-communicable diseases could contribute to andropause. Now, LH levels are high in athletic menstrual cycle because the body kind of constantly wants to put the female into a phase where more levels of estrogen, let's say, are being produced, and under this LH environment of estrogen, let's say, and progesterone, the female it's more balanced. Stress would lead to estrogen due to the oxygen saturation, as well as the elevated cortisol levels, which both of these contribute to a toxic environment for a newly formed oocyte. So exercise something additional in regards to exercise. So females that are exercising from early on in puberty, they will go into puberty much later compared to their peers that are not exercising. Also, exercise, it doesn't necessarily mean that it will lead to females. So yes, exercise could prolong the ovulatory cycle of the female. So because certain cycles could be anovulatory, this can mean that a female that exercise will reach menopause much later. Okay, now low estrogen because of low fat, because estrogen, in order to be secreted, it needs the mobilization of fat. That could come in a variety of forms, such as cholesterol. If that individual does not have any cholesterol, then the body has nothing else to mobilize. That's why low fat equals sometimes low estrogen levels. So if you allow me, and if you have any further questions, let's try and move on to our next PowerPoint slide and try and keep your questions relevant to your teaching objectives. I mean, it's very nice to know also some additional information, but I don't know if I will have the time to cover all of your questions. Okay. Now we're moving to the next phase of the female reproductive cycle, in which we're talking now about the release of our secondary oocyte in the peritoneal cavity. Now, during this phase where we have a mature oocyte, we also have the influence of our hormones such as progesterone that will aid the propelling of the oocyte to the uterus for implantation. So after the oocyte is being released, the fimbriae that you see over here, which are finger-like projections in the fallopian, in our fallopian tubes, will propel the oocyte into the uterine tube for fertilization. Conception has a very small window, approximately from 12 to 24 hours, during which, if the oocyte is not fertilized, it will it will eventually die. So having in mind that the sperm typically survives for 48 hours, it gives us a window approximately after the mid-cycle of the 14th day of, let's say, 72 hours more or less, or 76 hours more or less for conception to occur. So 4 days in total. So in the occasion that the fimbriae are not able to propel my newly formed oocyte into the peritoneal, into the fallopian tubes for fertilization, then we could be talking about ectopic pregnancy, in which the oocyte remains within the peritoneal cavity, eventually with the fetus forming and feeding from the rich vascular supply of the digestive system. Now, this could lead to life-threatening scenarios for the female, and it is particularly recommended that the pregnancy is terminated. Otherwise, we could be talking about a variety of other maternal complications. So for the sperm to be transported into the female oviduct for fertilization, a variety of factors need to be in place. First, the cervical mucus should be easily penetrable. We should have a a big amount of sperm that are being churned into the uterus for fertilization so that they can break the corona radiata of the secondary oocyte. And in addition to this, we need to have the presence of prostaglandins in the release present in the semen that eventually during orgasm, all of these aspects are gonna churn the sperm into the female oviduct for fertilization. It is important to note that ova are not particularly passive when it comes to fertilization, due to the fact that they also release calcium relevant pheromones or messengers, which they're gonna lead to a variety of intracellular changes inside the sperm that is also gonna lead it to start swimming towards the oocyte. So in fact, these particular receptors that are being present in the sperm, they're olfactory receptors, and they respond to the calcium that is being released by the oocyte. So in order for fertilization to occur, not only, as we said, we need a big amount of sperm that will be surrounding my oocyte, but we also need the acrosomal enzymes to start functioning. So in order for the sperm to penetrate the corona radiata that we see here, that it's basically a zone of granulosa cells, the sperm needs to start secreting for some form of digestive enzymes. As it passes the first phase, then is also gonna fuse into ZP3 receptors that are present in the corona radiata in the zona pellucida, and by fusing to these specific ZP3 receptors and the release of acrosomal enzymes, the sperm nucleus is gonna enter the cytoplasm of the ovum. As it enters the cytoplasm of the ovum, it's gonna activate cortical granules for the release of calcium surrounding the zona pellucida of the oocyte, eventually calcifying the outer region and inhibiting any additional sperm from entering the ovum's cytoplasm. During this phase, we have the release or the formation of microtubules that eventually are gonna fuse the genetic nucleus of the sperm as well as the oocyte. Now, after the formation of these tubules, we're talking now about a zygote. Now, a zygote is basically it consists of 46 chromosomes of both the female and the male gamete. And during this phase, we're gonna have the proliferation of a variety of cells that are gonna lead to the formation of a solid ball called morula. Now, the morula is gonna be propelled from the site of fertilization all the way to the uterus, where it's gonna be placed for approximately 7 days in order for the blastocyst to start forming. So progesterone has primed this environment for mammaliform morula, filling it up with glycogen through which the morula will be feeding over the period of the next 7 days. And it's also, under the influence of progesterone, is gonna relax the female oviduct for the propelling of the zygote down into the uterus. After this phase and after the blastocysts have formed, then we will have a Velcro-like formation not only on the blastocyst, but also on the endometrium. Thus, this adhesive surface over here is gonna provide a fertile ground for my blastocysts to land and slowly for the cannibalistic action of the blastocysts to start forming in the endometrium. During this phase and of implantation, we will have a minor edema that will be forming, being driven by my blastocysts, which is gonna mobilize proteins, glycogens, as well as the formation of other blood vessels to start being attached to the blastocyst. Now, during this phase, of the formation of the trophoblastic cells, eventually these trophoblastic cells are gonna degenerate and they will form what we're gonna be calling later on the fetal portion of the placenta. Now, the fetal portion of the placenta is gonna be formed by the multinucleate syncytium, which has been caused by the degradation of the variety of cells that are present at the endometrial wall. Eventually now, the trophoblast, now being called the decidua, has implanted itself into the endometrium, and now we also see a zone of endometrial cells engulfing or surrounding the newly formed life. From here, we're gonna have the formation of the fetus. We have the amniotic cavity over here. We have the yolk sac, and eventually through this place, we're gonna have the formation of the embryo. So the local release of prostaglandin by the endometrial cells is gonna lead to the vascularization of this region for the newly formed fetus to start feeding from the endometrial wall. So as the trophoblast continues to digest the surrounding residual cells, we're gonna have an increased amount of energy until the placental develops. During the trophoblast, we also have the release of fast ligands and tryptophan, which are modulating factors when it comes to cytotoxic T cells. Now, cytotoxic T cells could jeopardize the life of the newly formed fetus. For this reason, fast ligands goes and attached itself to cytotoxic T cells, eventually leading them to undergo apoptosis. And on the other hand, tryptophan, which is being released, it also leads the cytotoxic T cells to get immobilized. In addition to this, we also have an increased accumulation of regulatory T cells that suppress the cytotoxic T cells, keeping them in low quantities. So as time passes and the fetus starts forming, we do not need any more the corpus luteum of pregnancy, since the placenta has now starts to forming. As soon as the placenta starts forming, we have an increased number of estrogen, progesterone, as well as human placental lactogens. The placenta, as we talked about before, has been formed by the multinucleate syncytium during the trophoblastic implantation and the decidual tissue of the endometrium. So the placenta now will support the parasitic tissue, the parasitic fetus, and is going to become the lungs, the respiratory system, as well as the kidneys of the child. During this phase, we have the exchange of oxygen, water, as well as other aspects for the nourishment of the fetus. However, estrogen and estrogen release by the placenta cannot happen without the help of the fetus. So the placenta functions also as a temporary endocrine organ. So it's important for secreting the human chorionic gonadotropic hormone, estrogen, as well as progesterone. It supports and enhances the growth of the fetus, and it coordinates the timing of the parturition. It prepares also the mammary glands for nourishing the baby after birth. So beyond the support of the fetus when it comes to nourishment, it also coordinates the timing of the parturition, basically through the release of specific hormones. One of these hormones is oxytocin, which is going to prepare the endometrial wall as well as the myometrium, as well as the uterus, for the rhythmic contractions that will happen during parturition. We also have the release of cortisol that happens specifically through the adrenal glands of the fetus, that is particularly important for the maturation of the fetal lungs. And beyond that, we also will have the proliferation of the mammary glands, as well as the lactigenous glands, that will nourish the babies under the influence of estrogen and progesterone for after the fetus is being released. During this phase, and as soon as the newly formed life implants in the endometrium, we have the release of human chorionic gonadotropic hormone. Now, the human chorionic gonadotropic hormone basically extends the life of the corpus luteum, now called the corpus luteum of pregnancy. Now, HCG has the same function as LH. The reason why the human chorionic gonadotropic hormone is important is because that if progesterone and estrogen levels are not sustained at high levels, then the endometrium is going to lose its rich supply from oxygen due to the presence of a variety of vessels that have been created under the influence of estrogen and progesterone. If we do not have the influence of the human chorionic gonadotropic hormone, then we do not have a successful pregnancy. And this is why new measurements immediately at the first month of HCG tells a gynecologist of whether or not a pregnancy is successful. So under the influence of estrogen and progesterone, we also have the endometrial tissue being thick and pulpy, allowing a further buildup of nutrients for the newly formed life. So the human chorionic gonadotropic hormone occurs peaks usually after 60 days, and after its peak and as soon as the placenta is being formed, then it is no longer needed and it slowly starts decreasing. So HCG is being secreted by the fetal portion of the placenta, which it also plays an additional role, as we're going to see later on, in the maturation of the fetus. So as we said, during this phase, the placenta cannot produce the amount of estrogen that are needed for the sustenance of pregnancy. Thus, it cooperates with the fetal adrenal cortex by prompting the anterior pituitary of the fetus for the release of adrenergic corticotropic hormone during which we have the release of cortisol as well as dehydroepiandrostestosterone or androgens. So through this formation and through this communication, we have the conversion of androgens into estriol. Now, estriol measurements in maternal urine tell a gynecologist of the viability of the fetus, because if the fetus has not formed at an adequate level for the production of estrogen, then the pregnancy is most likely not gonna carry on. Now, during this phase, and even though the placenta does produce enough amounts of estrogen, it also needs the conversion of cholesterol to progesterone. Now, this can be facilitated by the placenta without the help of the fetus, as we've seen during estrogen. And as soon as the progesterone levels rise, we do know that the placenta is also increasing in size. When it is time for delivery to occur, we start seeing the slow death of the placenta. So the calcification of the placenta later on during pregnancy also signifies more or less the approximate delivery. So by examining regularly the progesterone levels, a gynecologist knows whether the delivery is going to be premature or the child is going to come later on. Because if we do have a healthy placenta, then we still have the space for the growth and the nourishment of the child. So I see that you may have some questions, maybe.
Doctor, please would you go over the sperm penetration of the ovum again?
The sperm penetration of the ovum.
Mm-hmm.
Okay, yes, absolutely. Glad to. So, as soon as fertilization occurs, where we have both the sperm and the secondary oocyte coming together, then we're gonna have the fusion of the two nuclei, which is gonna lead to the formation of the zygote and the second polar body. Now, during this fertilization that we see over here, we're gonna have the acrosomal release that will start breaking down the corona radiata. It doesn't necessarily mean that only one sperm is gonna break out this zone. Multiple sperms are usually needed for the breaking of this zone over here. Now, as soon as these acrosomal enzymes are being released, and we have the attachment, the successful attachment, let's say, of the sperm to the ZP3 binding sites, then I will have the second acrosomal enzyme release, which is gonna break down the corona radiata. Sorry, which is gonna break down the zona pellucida. As soon as the zona pellucida is broken down after my sperm is being attached to the ZP3 binding sites, so in order for the release of the second acrosomal enzyme, the sperm needs to bind at the ZP3 binding site. So these glycoprotein receptors over here, they're gonna allow a safe passage of the sperm nucleus in the ovum cytoplasm. What's gonna happen later on, we're gonna have the activation of cortical granules, which is gonna lead to the calcification of the zona pellucida, eventually stopping any other enzyme for penetrating the ovum. During this phase and after the successful passage of the sperm of the nucleus, we're gonna have the cross-linked molecules in, we're gonna have the formation of the microtubules at the nucleus of the ovum, which is gonna lead finally at the second meiotic division. This second meiotic division is gonna give me the zygote. Over a period of some days, the zygote is gonna be propelled under the influence of progesterone, which is gonna widen the passage of the female oviduct along with a variety of other peristaltic contractions into the uterus, into the uterine lumen, where the morula is gonna develop into a blastocyst. And also under the influence of progesterone, I'm gonna have an increased glycogen release that is gonna feed the morula until it completely develops and it becomes ready for implantation. Does this cover the question?
Yes, doctor. Thank you.
You're very welcome.
There are some
Doctor, can I ask you a question?
questions in the chat window.
Okay, I see we have some more questions. Now, okay, the oocyte could get fertilized by, let's say, by a sperm. Now, it does not necessarily mean that the sperm can travel a variable of distances. It doesn't necessarily mean that the oocyte needs to always be present in the female oviduct for fertilization. So it could occur also as soon as it's being released without even passing from the fimbrium. So that's when we're going to have the ectopic pregnancy. It could happen it could also happen within the fallopian tubes. It could happen within the female oviduct. So depending on the ectopic, there are variable occasions of ectopic pregnancy. It does not only mean that it will implant in the digestive tract. So estrogen is being secreted by a synergy of the placenta and the adrenal cortex of the fetus. If the adrenal cortex of the fetus is not mature enough, then it cannot produce androgens that will be converted to estrogens. So the human chorionic gonadotropic hormone the human chorionic gonadotropic hormone is going to stimulate the anterior pituitary of the infant for the release of ACTH, and the release of ACTH is going to prompt the adrenal glands for the release of androgens as well as cortisol, as we're going to see later on. This is going to lead to the formation of estrogen. So there's a communication between the placenta and the fetus. So let's move, all these questions that you have now will potentially get solved in our next lecture.